this is I'm proposing to Dr. Lokman, because we need to make use the time we spend here. We want to utilize the time so that it can benefit the students as well as the researchers. We want to highlight what other people are doing in the lab, or maybe what they are trying to do in the future, or what they have done before, and still they want to share knowledge. They are more than welcome to share, right? But this is kind of a kickstart. We just want to give like the first the first talk that can relate, okay, with our expertise and a niche area. So today I'm proposing my first science talk, the first series, using stem cells to understand human organogenesis. So we know that stem cells is a single cell, okay, it can develop into many other cells. Okay, so if you can see here, you have the nerve cells, you have the smooth muscle cells. For example, you have the liver, you have the blood cells, you have other cells. Okay, I'm not sure what they are, but they are different specialized cells. They are different types of cells capable of doing a certain activities. Okay, so this is the power of stem cell. They can be differentiated into a specialized cells. They are very powerful cells that they can, they can self-renew, and at the same time they can also differentiate it into other morphology. So it starts off with a life story. Okay, this is you and me. I mean, this is you. Okay, made up of two components. Okay, the egg and also the sperm. And what happens next? You will form two cells. Okay. As the growth develops, you start to produce 4 cells, and after that 16 cells, and after that you reach the blastocyst stage. This is the embryonic stage, where you can find the inner cell mass. Okay, the cells is inside. Okay, let me show here. But the one here is the inner cell mass of the blastocyst, and that is where you can find the stem cells. Okay, that is where they reside. Most of these cells are these embryonic stem cells, and they are pluripotent. Pluripotent means they are capable to differentiate into many other organs. Okay, they are not multipotent. Multipotent have a lesser capacity to differentiate into other cells. Okay, pluripotent is quite early, and obviously we have the totipotent cell as well. Okay, totipotent is what's what Dr. Lokman is doing, like a spermatogonial cells is the totipotent. It's very early here, top here. Okay, mine here. Okay, so that's the difference. Yeah. Okay, then it makes you, once you be delivered, then they will start to grow and become bigger. So stem cell have two main capabilities. They can self-renew. It means they can duplicate and copy their same information and then pass it, become a daughter cells. On the other way, the stem cell also have a powerful characteristics that they can differentiate or become a specialized cells. For example, the somatic cells, cells of your body, like muscle cells and nerve cells. You have liver cells, hepatocyte. You have kidney cells, you have neuron cells, okay, you have neurons, brain cells, yeah? So this is the type of cells that can be produced or can be generated by stem cells. So this is another capability to illustrate this. Whether you have the stem cells or not, it still can copy and also differentiate, all right? It can mix into 2 identical stem cells. It can mix into one single stem cell and the other one into specialized cells, okay? Or they can just become specific or committed to produce only identical stem cells, and also the other way around, okay? So this is about the self-renew capacity and also differentiation capacity of the stem cell. You begin with the 1 stem cells, and then at the end, you can see it keeps on producing their own cell. Okay, by time as you develop, you still have these, unique stem cells in your body. But at the same time, the stem cells have the capacity to become a specialized cells of your body, and they can carry out certain function. All right, so you have the muscle cells, you have brain cells, you have whatever cells in the body. Yeah, so they have two different functions. For the cells that can self-renew, they maintain their characteristic, but for the specialized cells, they can be differentiated into other cells that is important, especially if you have any mutation or maybe have some like cancers or certain disease, they will replace those faulty cells. Okay. Where they can be found? Yes, again, this is the embryonic stem cells. They are pluripotent cells. They can be found in the blastocyst stage of the embryogenesis or embryonic period. All right. So there are different types of stem cells. We begin with the embryonic stem cells. This is the inner cell mass. Okay, the outer layer of cells. Profactoderm is not included. All right. So this is the one that we have taken. We isolate this inner cell mass, the ICM, and then culture it in vitro. And after that, we can decide whether this pluripotent embryonic stem cell, whether we want to direct them to what organs. Okay, we have to know that we have the three germ layers. We have the endoderm, we have mesoderm, we have ectoderm. So from this starting point, we can mediate the expression of certain genes. We can stimulate certain genes inside the cells, and then they will drive or differentiate towards the certain organs. Okay, from what lineage? Either endoderm, okay, endoderm or mesoderm. Okay, so this is what we've done. What we will do inside the lab. Okay, we will provide the nutrients for the stem cells. What they can do, they can be differentiated into some other cells. Again, the challenges, you know, if you are about to use the embryonic stem cells, they are open to the ethical considerations. Different countries have different regulations in allowing scientists to use the embryonic stem cells. Okay, some controversial issues like the religion, like Islam, Jews, and Christian, they have a different way of allowing at what stage is embryonic stem cell is allowed to be used in the lab. Alright, so we have a different or ethical religion issues regarding the use of embryonic stem cells, because some of them consider this is a live being, okay, from sperm and also from the egg. Thus, if you throw the cells, if they die, it means like you are killing the people, okay, the human being. How about Islam? So you have to find out. I think Afiq have the answer. Maybe you can ask Afiq. Alright? So you want to, you know, stimulate the cells. This is the stem cells. You want to stimulate whether, you know, you put the recipe, certain recipe to make the skin or neurons, the blood or liver. Okay, skin, ectoderm, right? Neurons, maybe endoderm, maybe blood, I'm not sure. Liver, mesoderm, okay, whatever, okay? I forgot about the three lineage. Okay, so we decide what will be the lineage that we want. Okay, but the tissue stem cell we can find in any of these adult cells. Okay, you can find from this, you know, somatic origin. Okay, how about the multipotent? Okay, it's include blood stem cells, and those found in the bone marrow, they are not pluripotent. Means they are almost committed to a certain function. So they have a reduced capacity to make another organ. Okay, so they have the only capability to differentiate into that, you know, red blood cells or white blood cell, platelets, all those sorts of cells. Currently, I mean not currently, it's about like maybe already 15 years since the first time the scientists found the discovered the induced pluripotent stem cells. So what's the difference? Induced pluripotent stem cells is those cells that have been exogenously stimulated with a certain reprogramming factors. We call it Yamanaka factors because in 2006 the scientists who discovered this, they start with stimulating a certain 4 different set of factors or genes inside the animal, and they prove that they can reverse the process. They can reverse from adult cells into the embryonic stage cells. Okay, they can reverse the process back to the pluripotent stage. Okay, how amazing is that? And this genetic reprogramming, okay, you take like skin cells, and after that you make it the iPS cells. Alright, with the 4 reprogramming factors, and it behaves like embryonic stem cell, and they are pluripotent cells. Once we have the iPS, the reprogrammed cells, we can again differentiate them into any mature cells, any adult cells, and these bypass the ethical consideration when we are using the embryonic stem cells. Okay, so some religion, they don't allow the use of embryonic stem cell. Alternatively, we can use the iPS cells. Okay. All right. So this is just example in life. Okay, when we do the reprogramming overnight, when you stimulate with the reprogramming factors, like when you take skin, okay, a biopsy. After that, you put it in, isolate the cells into the culture dish, and after that, you give a certain nutrients for them to grow, and then you start to add up all these 4 differentiation, the reprogramming factors. Okay, the 4 reprogramming factors reverse back to the pluripotent. Okay, you can say from the adult stem cell to the pluripotent stage, and after that, you can convert or differentiate them into any tissue of your body. For example, if you take from the skin, you can make the bone. Okay, from the skin, you can make the skin again, or you can make the skin into the muscle. Alright, so that's the concept behind it. Okay, so this is just a jargon. Maybe some of you already experienced or know about this one. And also we can have cloning too using the stem cells, reproductive cloning. You can make 2 identical individuals, a total absolute copy of you. But however, this is very difficult to do and it's illegal. However, what we can do in the lab is what we call the molecular cloning. We use to study what are the function of the genes, what the roles of the genes. And this is already a routine in the biology labs, right? So you have known about this reproductive cloning. I think Dr. Lokman also is expert in this stem cell technology. But in brief, we can say that you're taking the nucleus that contains the chromosome, the 46. Okay, you combine with the what we call enucleated cells, and after that you will make your own clone. In the lab, we get used to the molecular cloning where you take out the DNA of the nucleus, okay, take out DNA out from the nucleus, alright, and then you take the cell too, and then you combine them together, and then they can make a new cells, okay, express these 2 different genes, alright. So you can edit the genome, edit the genes of the organism too. In terms of the application, you can study the roles of certain genes. You can use the molecular cloning to remove a gene to see whether anything works differently. Like normal mouse embryo, you want to see whether, after you silence or you knock out a certain gene, what will be the outcomes, alright? So genes is involved in the giving the eye its color. Maybe they change color now or colorless. You use the reporter gene as well. You want to add a certain genes that show us when another gene is working. Okay, gene is active is in blue areas only. Okay, you want to see the expression of certain genes, whether it's expressed in the brain or in the stomach, on the gut, or in the liver. You want to see, right? Certain gene. For the lineage tracing, also it's important for scientists to use this technique in their activity related to the stem cells. Alright, so I think this is one of a kind of summarize. Before we produce, maybe it looks like very easy from stem cell, become a specialized cells over there. But in the middle, okay, in between, there must be a committed progenitors. I mean, this is the earlier or intermediate cells that can give rise to the final cells, all right, the terminal cells. So it have a various cascade or coordinated pathway for them to become a specialized cells. And those committed progenitors, they are multipotent, okay, because you have reduced the capacity from pluripotent, multipotent, and then become the specialized cells, right? So this is the process that how we want to generate an organs or tissues or another specialized cells. This is another example. In Malaysia, we have various institution currently active using hematopoietic stem cells, and also it has reached the therapeutic stage. However, cells related to the brain cells like neural stem cells and other organs are very limited, and still under the we did not really go to the clinical trials yet, but some of the neural stem cells start to being implemented inside the preclinical studies. So this is a type of, this is example. You want to take the cell from the brain, all right, which is the stem cell, or you can take the adult cells from the brain, convert it into the iPS, and after that you will induce with a certain practice and to form these progenitors. After that you can make all those cells at the end there. Okay, gut stem cells as well. This is like the most famous. Brain, hematopoietic, and also the guts, as well as the mesenchymal for the bones. Okay, you want to make a cartilage, you want to make the bone, fat, everything is possible. And obviously this is inside, I mean, in the research level. It's not really go into the clinical yet, okay? And it's limited for certain countries only. Stem cell niches, this is the microenvironment surrounding the stem cell that provides support. They are accessory cells that can supplement the stem cells so that they can grow, okay? They can provide certain proteins or molecules so that the stem cell can keep on growing and develop inside the culture. It's either direct contact, or you want to use small molecules, or you can have the help from other cells so that the stem cells can grow. Okay, that's the first part of the basic introduction about the stem cells, IPS, about the early knowledge. Now about the research, how we can use our knowledge with the stem cells to treat human. I mean, we need to solve a certain issues. You know, in SDG, for example, goal number 3 for human like well-being, something like that. So we need to improve in terms of these diseases. So you need to come up with strategies how we can treat certain human ailments. Okay, one of the things that we can do using stem cells is by manipulating the stem cells for the benefit of the humans. And in theory, we can create a disease model inside a dish. Okay, in the lab, we can create an organ which is similar to human models so that we can start to test any drugs, something like that. Yeah. So my focus today is in the field of the kidney diseases, where you see every day lots of people spend thousands just for dialysis, hemodialysis, or they have to wait a longer period of time, and okay, it can goes up to years before they can be, find the donor for the organs. And this is very timely cost, consuming lots of cost. And again, in the lab, we are doing research relating to the IPS, how we can make a new kidney. Lots of manipulation that we can do by stem cell technology. You can start with the IPS, obviously, or embryonic stem cell, and after that you can study a certain pathway that they can differentiate into the final or the terminal organs or tissues. You can do some genome editing if you want to test what happened if you cut a certain faulty genes or cut certain healthy genes. You want to differentiate them for another or therapeutic purposes or for the drug discovery, you may do that using the iPSCs. For these diseases, I'm interested in understand how the kidney is being regulated or is developed inside our body since the embryonic stage. We have these issues with the kidney. During early stage of development, you can see the mesonephric duct and metanephric mesenchyme, they must attach together as we grow from the first trimester, during the gestational period. So this is important. Okay, the fusion of mesonephric and metanephric mesenchyme is important for you to make a healthy or normal kidney structure. If anything happens, for example, mutation, mutation can occur if there's any damage or any issues during this fusion. You can see inside the kids, they will be born without kidneys, or some of the kidneys become cystic. Some of the kidney become hypoplastic, very, very small in terms of the structure. There are various of chronic kidney diseases related when during embryonic development the mesonephric duct cannot fuse with the metanephric mesenchyme. And this is my talk for today. My hypothesis is that I can use the blood samples and then differentiate them, reprogram them into iPS, and from the iPS cells I want to make a new organs, a mini organs we call the kidney organoids. So what I did was isolate the blood samples. I took the sample from the patient that have the disease just now, that have the cystic kidney disease, and then together with their healthy parents, I want to see the comparison. Every time you have the control and also the disease one. So both of these cells being isolated from a peripheral blood mononuclear cells. I spin them down and just take this what we call a cloudy substance there, and that is where the mononuclear cells are. So they have the information of these stem cells, and after 1 week in the culture I put all these 4 different reprogramming factors, and after 2 weeks we can see that the iPS is generated. For the whole process it can take around 100 days. Very time consuming, but nowadays we have an alternative to make it short. And this is the type of example of stem cells. Some of them are quite fragile. They easily detach from the culture plastic. So it's important for you to maintain the health of the stem cells quite every day. The media changing will be every day, instead of every 2 days. So this is what happened. From the blood you can make a iPS, and the experiment onwards I will take these at the end there, the iPS then, and differentiate into kidney organoids, right? My concern or my focus is the HNF1 beta. HNF1 beta is the transcription factor. It's the homeodomain containing superfamily of transcription factors that can regulate the normal healthy kidney development during the embryonic stage. In other words, the HNF1 beta gene functions to control the normal kidney development, right? So if you have mutation in this HNF1 beta gene, you cannot fuse the mesonephric duct with the metanephric mesenchyme just now, and it will lead to the cystic kidney diseases, right? So this is just example. We have 9 exons of this gene, HNF1 beta gene, And I also do some transcript, expression of the transcript for exon 2. This is the famous one. It contains the missense mutation, and also the exon 9, those quite near to the 3' untranslated region, the 3' UTR there, just to make comparison. But there's a very few papers suggesting that the mutation occurring there. Okay, the most famous one occurring quite early, which is about the first 4 exons of the genes. And based on the expression, okay, the red one means the faulty one, the blue one means the healthy one. You can see in terms of the expression is quite similar. I can see that the expression of the faulty cells is higher as compared to the healthy one. It does mean that something happened in terms of the HNF1 beta expression inside the faulty cells. They are higher. So I can conclude that the iPSC lines is successful. We can manage to differentiate them. And after that, once we have the IPS, we want to modify and stimulate the differentiation into this, this here. Okay, so this is the mesoderm. The kidney is the mesoderm lineage. Okay, you can also use the cell IPS just now, okay, into the skin, neural, or pigment cell, which is the ectoderm layers. You can go to the endoderm layers by manipulating these pathways. Okay, this is the molecules that is important. If you want the cells, the iPS cells to become a kidney, you have to play around with these factors. BMP, okay, these are all the growth factors and also signaling factors that can drive the differentiation into the specific organs. So once I have the cells, IPS just now, I culture them, okay, in monolayer culture, which is in 2D for the first 7 days, okay, 4 days and 3 days there. Once it's reached the confluency, I will put them into a test tube, not test tube, this is the appendix tube, spin them down around 200,000 cells. After that, I will put onto the special container, what we call it, inserts, and expose the clump of cells to the environment, the air, and also the nutrient below. So they will become bigger as they pass through the differentiation course. Okay, this is an example at the 2D stage, which is only monolayer up to max day 7. You can see they are confluent. After day 7, at day 7, I will pick them up and then dissociate them and mix into a ball of cells. After that, I will induce the cells into a kidney structure. And as you go onward, you can see the complexity. The structure becomes more intricate, and the histoarchitecture did express some of the important kidney structure. Okay, for example, this is the day 7 plus 9, means this is the intermediate progenitor stage. They are not mature yet. Okay, this is a bit more mature, where you have these NPHS1 factors or genes being expressed. That's like a glomerulus, which is the functional filtration unit for the kidney, right? And some of the other tubules related to the kidney structure. So if you look, in the cross section, you can see the histoarchitecture and also the regionalization. Okay, for this kidney or nephrons, you can see the tubules at the bottom, and in the middle you have this tubular structure, and at the top you have the glomeruli that functions to filter all the waste or the water, ions, and electrolytes. So if you compare the control and the mutant one, the control have a fixed structure and they are more healthier. But the mutant, you can see, is not that complicated. They are dispersed, not so healthy. So we would suggest that we hypothesize that these mutant cells that have the HNF1 beta mutation, they carry the genes, they carry the faulty genes, and the structure makes the cell become hypertrophic or become bigger. And that correlates with our assumptions that HNF1 beta leads to the cystic features, right? So this is just an example, and we did some of the staining. If you compare the mutated one, okay, unaffected is the healthy one, the mutant, okay, 172171 is the one that have the mutation. CDH1 is the gene that's good for the epithelial structure. The SYNPO is the synaptocoding marker for the glomerulus, and HNF1 beta is the transcription factors. You can see when the faulty genes is expressed, you can see there's a very high nucleus or very high staining intensity for HNF1 beta. It means that could be something happen inside the faulty genes, okay, inside the faulty organoids. So we are able to differentiate these kidneys into IPS into kidney organoids, and we can compare that the mutant is totally different from the healthy one. Okay, so this is one of the examples. I want to highlight that when I stimulate the cells or the organoids with a certain chemicals like the cyclic AMP, just to check whether they can respond or not, like the human cells, and they do show some effect. Okay, this is recapitulating what we have seen in the human model. So I don't want to discuss a bit more on this because it's going to involve the second messenger, the 6-CAMP, and also some of the signaling pathways. Alright, so I just like to recapitulate that we are able to do this one, okay, in the lab, and we are still under the research stage, and we are about to push this forward. Okay, what's next? We want to differentiate these organoids that can improve the vascular system. Maybe we want to integrate more blood vessels inside it so that more oxygen will come and supply the kidneys so that they can do the filtration function. Second one, we can optimize the differentiation protocol in such a way that they can make the ureter. I mean, if the organoid able to make their own urine, we can pass on the ureter into the dish. Okay, and we can check the protein that being expressed or the chemicals available after certain time points. We can also implement the gene editing strategies to create a perfect control. We can do transcriptomics research using the NGS to uncover the variability, heterogeneity between cells, and what are their pathways, their regulatory pathways, and formation of cysts in the glomeruli due to urine flow obstruction. In human with HNF1 beta, collecting drug is most affected. Okay, in the case that I've mentioned earlier, those kids that has been born with a cystic kidney, they have the ureter also affected. So they can pass urine, but then they are very in what we call morphology. The morphology is distorted. Okay, they require a quick transplantation. Thank you very much. I will stop up to that point for now. If you have any questions or suggestions that we can improve, I hope that this kind of giving a quick kickstart for us to continue for the next session, Science Talk 2 and also the Journal Talk. Thank you for your attendance and attention. Thank you.
if someone have a question to ask, can ask the doctor.
About the video on my presentation, yeah. So about the gene HNF1.
Yes.
It seems I want to say this overexpressed in this. Please show me the picture. The I think before that the one. The messenger RNA. This one. The response is overexpressed. It seems that this SNF1B is overexpressed in this abnormality in that. So it's associated this overexpression of these genes because of mutation. What kind of mutation? What kind of mutation? Because of like dominant negative or something like that.
It's a heterozygous. Heterozygous mutation.
So this overexpressed more than normal, right?
Yes. Based on the data, what I found that they express more than the normal level of
Is it associated with the formation of say something like that?
Yes.
So say if we try to block the expression.
That's, yes, that's what we want to do next. Actually, I don't have time to continue my study up to that point. So we want to, using the CRISPR, the gene editing, we want to block that one, or we just knock out that. This one quite early stage, because if we using the CRISPR here, if you cut it here, that you can create a stop codon, then the rest of the protein will not be translated, or no more proteins be produced. Okay, so we want to check, yes, as Dr. Prof. Soda said, this is what we found, but we want to really, confirm whether this is the direct effect of the gene or not. Okay, if we what we call knock out the gene, whether the same thing happens or there are new, insights happening after that. Okay, this is just, thank you for that.
We usually for the patient having this, we cannot do anything but just we just try to silence the expression. Hopefully they can be cooperating.
Yes. Yeah. Possible. Yeah. If we can block this action or the expression, maybe the expression level back to normal, and then the patient can live happily like a normal people. And maybe there's no more cystic features. Some of the proteins that functions to the cyst can be inhibited.
One more thing, related to the exon, you have 9 exon, right?
Yes.
So how you try it? How you ensure, is it only 9, or you just, it's like being standardized as 9, or you just try 9?
So this is what we found in the what we call in the genome. Okay, so the HNF1 beta gene only have 9 exons. Okay, some of them reported that already have 8 exon only, but the consensus is that we have 9 exons so far. Yeah, but towards the end of the gene, we can find that there are not more mutation. But it's not because of there's no mutation here, but because less limited research focusing on this exon. That's why we cannot find. It's not that this is more popular than this side. Okay, we need more research to uncover what's happening to that one towards the end. But if you have mutation in either of these exons, it still gives the cystic features in the kids. Regardless if you have the mutation in exon 9, you still have the cystic features, or the HNF1 beta still overexpressed. If you have the mutation in here quite early, also similar. There's no distinction between the exons.
And one more question is, when you do the XL here, in Angola,
Well, ideally the
have you done it?
well, at the moment we still contacting, looking for people or looking for the pediatrics that have this disease. I'm quite certain that in Malaysia we do have this one, because this is this HNF1 beta gene also contributes to the development of diabetic condition in the kids. So it leads to the maturity onset diabetes of the young, MODY, maturity onset diabetes of the young. It's also the co-outcomes or the outcomes of these genetic diseases. Okay, when you
mean DM1?
Sorry?
That one or that one?
I'm not sure, but this one, because obviously we cannot say that this is happening because of the cells in the pancreas. The beta cells is working well. They can produce a good amount of insulin, but I can say even though this is happening in the kids, but it's considered as the type 2 diabetes. Yeah, because the type 1 is purely because of the genetic factors involving the manipulation or mutation inside the pancreas. Well, the expression of HNF1 beta you can find is not only in the kidney, we can find a quite high expression in the liver as well. We can find the expression in brain as well, and some of the current research relates this gene to the autistic condition in the kids, and also it's worth to discover the relationship as well. So this is worth to be studied.
I think we can try the liver first.
Yes, that's preferably yes. We can do some expression studies like profiling of this expression. We can check inside the animal, and also at the same time we can try this in vitro as well, like in the culture dish. So both, to confirm our cell culture works, correlates with what we have seen in the animal. Yes, from
Suggestion, not suggestion. My two comments. First, because it's my first time listening to your, for me, a very wonderful presentation. And then I'd like to congratulate you that our university and in fact our company only donated to send you to Manchester University. That's the one thing. And then another one more thing. Do you have the thinking of foresee the compatibility? Now, when we studying this particular pathway in the so-called this ex vivo or in vivo, and then you culture in the dish, and then when you want to implement that in the real situation, more complicated human beings, for example, is it did you foresee the compatibility thing result?
Thank you, Prof. Muhammad. Yes, this is a very interesting question. So it goes back to our focus, because I think in the research cluster, we envision what we going to foresee in the next 10 years. How this research can support us, how this research can benefit the society, or we just doing this for our own gains, or it's not benefiting other people. So how do we tackle these issues? So the first thing is this stem cell is I can say it's profitable. At the same time, it's also important for people to learn. To derive the IPS is what we call is doable in the institution. Of course, if we manage to create our own IPS cells, then we can open more collaboration on that. Okay. The second thing is that in the long term, we are looking from this project. Once we have the disease model, the kidneys, the mini kidneys inside the dish, okay, from stem cells, we can integrate the facilities or the expertise that we have in IMOLEC, for example. People from the nanoparticles, people from the natural product, they can test the drugs. They can increase the filtration of this kidney, whether they are functioning or not, and then we can know that in the future, maybe next 10 years, we can use this system, I mean, as a model to, you know, recapitulate or to be practicable inside the scientific community in the future. Okay, that's the second one from the drug discovery or research, or you want to see the activity of the stem cells. The last one you want to see in the long run, like you need to create like a prototype for this one. So I've been thinking about integrating the, you know, a suitable condition that really mimics our human kidneys. So trying to create a 3D, what we call 3D bioprintable cells that can sustain these organs for a longer time. If we manage to sustain these cells using the 3D bioprinting, we make all the supporting cells or the niches very good, then we can sustain this, I mean, for the clinics too. So, I mean, in the next 10 years, maybe it's quite ambitious, but if we can provide the foundation for us to show to people that this is at the lab, the research, we able to test like lots of areas or lots of manipulations or modifications that we can do to bring this forward. Other than that, lots of other countries are using this biochip. They want to create a single organs in a chip. So whenever you put some drugs, I mean for drugs or chemical, then you can see, you know, expression of maybe 100 genes at one time. So it's for the high throughput screening. So instead of testing one compound, so maybe you can test like 100 at one time. So it can give a big data for us and can save time too. So these are, you know, the disease model. This is very important. Maybe some other people in Japan or in Korea are doing the similar thing, but we want to create the facility and we have our own facility and expertise to do things. We don't need to, of course we have to refer to them, but we have to try it first. Okay, if this works, okay, I mean slowly, eventually they can be implemented and can help, I mean, to move this technology to the clinics. People are doing, you know, stem cells and you know, cell culture is very, you know, prospective and also quite important for the countries. I mean, before we move to the clinical trial you know, stage. I think that's what's my view on the question.
Because why I'm asking now, because who knows that in 10 or 20 years, if I live longer, I'm the Prime Minister, so I will support you. Okay, I will support you.
But to be taken advantage of now, first we got the ICS meet in
I think as a start, we cannot start, of course we cannot start with the human, because you try to take a mouse embryonic stem cell and try to derive it, you know, in the lab, and, you know, just manipulate that one and see any compounds. Once we have reached, you know, a certain, you know, satisfactory you know, level, then we can move to the human. I think now is the animal study should be fine.
even if you have the line, then you mean animal line, we even can sell it.
Yes.
And have different lines for that. For IPS, for embryonic stem cell. So we can start with that.
Yes, but that's the thing. Obviously, I mean, preferably for the stem cells, especially dealing with the IPS, like either it's mouse IPS or mouse embryonic. If you start to, you know, sell it, then we need to observe, like record like lots of things, make sure that this is, you know, safe for the I mean, from any aberration. Like maybe you will have the chromosomal abnormalities. You have some mutations in the stem cells itself. So you need to be very careful before you sell any products. They should be safe and free from any mutations. I mean, all those 23 pairs of chromosomes free from any mutations. We can do karyotyping to confirm that and send it to the cytogeneticist to confirm our derived lines are safe or not. message really for today as a kids.